Hi, this is Miss Litton, and this is my wonderful Period 4 Honors Bio class. Say hi.
Hi.
And we are down, how many did we say we were down? 8. So we're down like 22.8% of the class. And so because we're concerned that you're missing class today, we are recording today's lesson for you. So hopefully you are a participant. Does anybody want to give a shout out to their missing bio buddies? Go ahead, shout it.
Mark. Sophia.
T-Bone's now in period 2. Be aware. All right. Now, let's go through. The first thing I want to do is paint kind of a picture of where we've come from on this. So you're thinking about this as you're preparing for your exam. The first thing we started talking about in this unit was chapter 5, right? And chapter 5 was all about cell division. And there were 2 types of cell division. Can you review those real quick with your bio buddy? What were the 2 types of cell division? Did you say my arm, my leg, mitosis, and myosis? And they have 2 different purposes, right? Mitosis is for growth and repair. You're making clones of cells. So if you have a cell with 10 chromosomes in it, how many daughter cells are you going to make? 2. 1 cell needs to review, right? You're remembering this? Got a test coming up? Okay. So you're going to do mitosis. Remember prophase, then what? Metaphase, anaphase, telophase, cytokinesis. Good. You're going to make 2 daughter cells. And if your parent cell had 10 chromosomes, how many chromosomes will be in your daughter cells? 10. Same, right? Keep it the same. All 10 chromosomes will replicate. They will line up single file during metaphase, and the sister chromatids will separate, and you'll have 10 chromosomes in each cell. Yes? Okay. Meiosis is different. What are you getting ready for? Sex. So you still are going to have the S stage of interphase. You're still going to have all your chromosomes replicate, right? But in the very first line up in the middle, in metaphase 1, the homologous pairs line up. So mom chromosome, dad chromosomes are lined up. And in anaphase 1, you separate the homologous pairs. Now you have 2 cells to do meiosis 2 in, and then you're going to separate the sister chromatids. So you make a total of how many cells? 1, 2, 3, 4. And if your parent cell had 10 chromosomes in it, those 4 cells are going to have how many now? 5. Because you're going to get down to the haploid number. Okay so far? If you're not okay with anything I've said, because I haven't even gone into the deets on that, then I would recommend that you jot that down as a note to yourself in your practice problems that you need to really relook at that. All right? Okay, next thing. Once we talked about mitosis and meiosis, because meiosis is for the purpose of reproduction, when you do spermatogenesis, which is the making of what? Sperm. You will make 4 sperm, okay? But when you do oogenesis, which is the making of what? Not eggs. You don't do equal what? Cytokinesis. You do unequal cytokinesis. So every division in meiosis 1 and meiosis 2, okay, but meiosis 1, you're going to have unequal division, so you're going to have 1 large egg that you're forming and 1 tiny little what? What's it called? I'm cold. This is an animal that lives where it's cold. Polar body. Remember that from your notes? You will make a polar body. Okay, and the reason why you do that is members of the fallopian swim team only bring with them their nucleus. Sperm only bring the nucleus. They're not bringing any nutrients. So when they fertilize, they're not adding anything to that cell, and that cell has to have time to get and migrate down the oviducts, down and implant in the uterus. So you do unequal cell division, so you have a 1 large nutrient-filled cell as opposed to 4 small cells like the sperm do. So cytokinesis 1 is uneven, cytokinesis 2 uneven, so you end up with 1 large egg and 3 little polar bodies that will degenerate. I'm going to recommend that you review that because I got a lot of bunch of blank looks on that one. Okay, so I recommend that you review that. Turn towards your bio buddy. Ask him, how'd you do on that quick little review? All right. Second chapter we talked about, chapter 22, was about development. So what happens once that sperm meets that egg and you form a diploid zygote? You do what? Cleavage, 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 and you form a what? Solid ball of cells called a morula. Then you pump in some salt, Who follows? Water, and you form a blastula, and a blastula is a hollow ball of cells. Okay, what do you call the cavity inside the blastocele? Let's let out the gastrula, and you form a layered ball of cells. At first, just the ectoderm and the endoderm, but then later the mesoderm. Put a pin in that, go back to the gastrula. What do you call the new cavity now that we've smished away the blastocele? Archenteron, good. Which will eventually become your gut. Yeah, your gut tube. Okay, what's the opening to the archenteron? That's your blastopore. You and I, that becomes our anus. Other organisms, it becomes their mouth. Good. Okay, back to those 3 layers. The outermost layer is called the ectoderm. The ectoderm will become your nervous system, skin and nervous system, right? The endoderm will become the only 2 systems that you can access through your gut tube. And what are those 2 systems? What'd you say? Respiratory and digestive. Everything else comes from your mesoderm. And when we even think, what's in the middle of our arm? What's in the middle of our arms? Bones and muscles. Okay, so those are 2 good examples of what comes from the mesoderm. Okay, then you have another stage. You had your morula, your blastula, your gastrula. Then the next stage you form the neurula, the nervous system. And that's when you have the infolding or invagination of the ectoderm to form your spinal cord and brain. Now, the tissue that got it to do that is your notochord, which is right below it, which eventually becomes your vertebrae. The notochord from the mesoderm tells the ectoderm above it, fold in. What is it called when one tissue influences another tissue? Induction. Influence induction. It's like peer pressure. This tissue is telling this tissue what to do. How does it tell that tissue what to be? It uses morphogen. Those are chemicals. And they cause it, once it starts moving, that's morphogenesis. The movement, the formation of organs and body parts is called morphogenesis. So induction is when one tissue influences another. They influence them using chemicals. What are those chemicals' names? Morphogens. When she does actually start moving to form those structures, it's called morphogenesis. Okay? Now, how can cells even be different when you all started being just one cell big? Okay? You were only one cell, yet you differentiate into other tissues. You would think you would just become a big ball of cells. But yet, they start to specialize. And why is that? Because whatever's in the cytoplasm is different. How does that come about when you're only one cell? Because wherever the sperm penetrates, the egg is going to be the what region? Anterior. And the other region is going to be the posterior. Yeah. Wherever the yolk is, that's going to be the ventral. And the other side is going to be the dorsal. That's vegetal pole and animal pole. And that sets up these chemicals in a gradient, or it's in ratios that are a little bit different, slightly different, which turn on other genes and start that determination pathway. That is chapter 22. Tell your bio buddy, do you need to review chapter 22 a little bit? Okay, you know it's not going to happen unless you actually make it happen, as in setting aside to review those 2 chapters, right? Okay, then that brings us to our current chapter, okay, which is, all right, so we know how to make more cells of ourselves to grow. We know how to make sperm and egg. We know what happens when sperm and egg get together during special hugs and you form a diploid zygote, and how those tissues develop. How do our genes, our DNA, influence what traits we have? And that's what we're talking about right now. And we already talked about it. We talked about genotype versus phenotype, right? Genotype is what your DNA is, and phenotype is how those proteins look when that's coded for, when those genes are coded for. We talked about doing Punnett squares, simple crosses, right? We talked about dihybrid crosses, right? And we could even do a trihybrid cross. Remember how we worked our way through that? We said do individual what? Punnets, and then multiply the probabilities together. Okay? Ask your bio buddy, how did you do on that when you did your practice problems? You got that down?
Same. Also, but special.
Okay? And now now we're here. We're going to level up one more time. We as scientists like to look at trend lines and to predict things. And that's what pedigrees do. Pedigrees are you looking at parents, their parents, the parents before them. You can predict about the children, and you can talk about how different traits are inherited. And so far, we've just limited ourselves to autosomes. Autosomes are everything except your sex chromosomes. Okay? So for me, my sex chromosomes, because I'm a biological female, my 23rd pair, my sex chromosomes are X and X, right? Females are X and X. So I have 2 copies of the X chromosome. A biological male, he is not XX. What is he? XY. So he has 1 shot at his X. Do you agree with that? Does it matter if that trait is dominant or recessive? No. He only has 1 X. He gets what he gets. Who gives him it? Who gave him his 1 X in all of his cells? His mom. Okay? Because his father gave him his Y. How many X's do I have? 2. Does it matter dominant and recessive for me for sex-linked traits? Yes. Because 1 X could be dominant over the other X. If he gets a recessive trait on his X, he gets that disease. He doesn't need a second recessive to get it. It's the only thing he's got. That's the only card he has to play. He has 1 X card to play and 1 Y card to play. Girls in the room, we have how many X cards to play? 2. Okay? So when we start talking about that inheritance, it matters whether you're a boy or a girl. Okay? When we talk about autosomes, the other 22 pairs, it doesn't matter if you're a boy or a girl. They are what they are. You're going to have your inheritance. Okay? And we'll review those and also some exceptions to those rules today. All right, so the first thing I want to do is on your group shared notes. Okay, so we left off at autosomal patterns of inheritance and a pedigree. Do you see that? Okay, so autosomes are any chromosome other than a sex chromosome. Any chromosome other than a sex chromosome. And both males and females are affected with equal frequency. Okay, in pedigrees, circles are female and squares are male. All right, so let's take a look at this pedigree right here. Okay, now this pedigree, they are identifying it as whatever you're inheriting. You know, if this is a disease or something, this is a recessive disease. If you look at the parents at the very top, mom has the disease, and I know that because it's colored in, okay? She's colored in, and she's homozygous recessive, so I know that's the type of disease that I have. Dad, why is it big A question mark for dad in that first parent generation? Tell your bio buddy, why is it big A question mark? So the only thing we know for sure are the ones that are colored in, okay? So we know those. Why? Oh, here's another reason maybe not to do cousin loving, right? Because we all carry with us a bag of recessive traits that maybe aren't so good. But if we, marry and have children with our sibling, okay, or our first cousin, we have a greater likelihood that some of those recessive traits are going to come up that we are not going to like, okay? So let's take these 2 relatives right here, these cousins, okay? How come they're big A, little a, and not big A question mark? Why did we know their second one? Yes?
Because they have children who are affected, so you know that they both have to have a little a.
So perfect. And that's how you analyze a pedigree, just like that. Because they have children that are affected, they must be heterozygotes for it. What was the odds that this child right here was going to get that disease? 1/4, right? If we were going to Punnett square these cousin Levins, big A, little a, big A, little a, do you agree? And the only one that was going to get that was right here, right? 1 out of 4. Do you see that? These two right here, they are carriers for that disease. Do you agree? And this one right here is the only one totally in the clear. Right? What can we say about this one, regardless of who it marries and has children with? What can we say about Mr. Blue? What can we say about their children? Tell me, everybody. Yeah, their children. It doesn't even matter who they're married to. Their children are not going to get it because the only thing they have to give is what? Big. How about our pinks? Could they end up giving this disease to somebody? Yes, if somebody else is a carrier or they actually have it, right? If they're a carrier or have it, they could give it to their children. How about our yellow? Could they give it? Yes, if somebody else is a carrier or has it. See how that works? So we can predict about them based on that we know it's a recessive disease, that's how that disease is passed on. So take a look at your notes for a minute where it says autosomal recessive disorders. Most affected children have unaffected parents. Most affected children have unaffected parents. Okay, case in point right here. Okay, neither parent had it, but it did show up in their children. Okay, and then number 2, heterozygotes have an unaffected phenotype. Could Blue please explain why number 2 is true? Go ahead. Heterozygotes have an unaffected phenotype. Because what kind of inheritance is this? Not a dominant disease, but a recessive. So if you're a heterozygote, you don't have it, right? Like if blue eyes was a disease, and you were heterozygous for eye color, big B, little b, right? Do you have the blue-eyed disease? No. But are you carrying the blue-eyed disease? Yes. Could you pass it to your children? Yes. Okay? All right. Now, then number 3, 2 affected parents will always have affected children. 2 affected parents will always have affected children. Slate, could you please explain that one? Why 2 affected parents will always have affected children. Go ahead, Slate, in 1.
So the unaffected is dominant, but if both parents are affected, that means they both have privilege. So no big N
Okay, hang on just 1 second. And you can see if 2 affected parents, right? If this girl marries this boy, all they have to give are little a's. So if you think of blue eyes as a disease, man has blue-eye disease, a woman has blue-eye disease, what's going to happen? All the children are going to have blue eyes, if you thought of it like a disease. Okay, that's what you can predict if you know how it is inherited. Yes?
I missed autosomal patterns of inheritance, number 3.
Oh, in a pedigree, you're so sweet. You didn't miss it. I didn't say it. That's on me. In a pedigree, females are circles, males are squares, affected individuals are solid colors. Affected individuals are solid colors. All right, let's look at some traits that are usually inherited as a recessive disease. The first one is Tay-Sachs. This one is very sad. This one has been in this Oak Park community. In Tay-Sachs disease, baby's born, looks perfectly normal, acts perfectly normal, starts progressing, starts developing, but then maybe 1-2 years of age, they start working backwards. They lose some of the motor skills they have. They cannot feed themselves. They can't hold their heads up. Their hair starts to fall out. They can have seizures, blindness, paralysis, and usually by the time they're 4 or 5, they have passed away. I mean, it's horrible because it's horrible because you think everything is fine with your children, and all of these children have passed away. You think everything is fine with your kid, and you're so excited, and then all of a sudden, no. And now let me tell you why it struck our community, maybe more than another community. Jews of European descent are more likely to have Tay-Sachs than non-Jews. Now, right now, sitting in your seat, could you have Tay-Sachs disease? No. How do we know that? You wouldn't be alive right now. You'd already have passed away. Could you be a carrier? Yes. You can actually go and get yourself tested to see if you are a carrier for Tay-Sachs disease. Now, here's the super sad part of it. Okay? The reason why it's more prevalent of Jews of European descent is because in Hitler's concentration camps, okay, that there were pathogens and there were diseases. And if you were a carrier for Tay-Sachs, you didn't have Tay-Sachs, but you were a carrier for it, you were more likely to survive those pathogens. Those pathogens couldn't take hold in your body as easily. So hideously, this is a form of artificial selection, because those that survived the concentration camps, survived to reproduce, are more likely to be what? Carriers of Tay-Sachs disease. And those that didn't have it were the ones that were more likely to perish in those concentration camps. Okay? This is also true of something called sickle cell disease. Do you know what sickle cell disease is? When the red blood cell, instead of being a biconcave disc, it actually sickles, like the shape of a sickle, like it's on the Russian flag or Soviet Union. Right? You know what I'm talking about? The sickle? Okay. So, if you are black, you are more likely to have sickle cell disease. And here is why. Okay? Because if you are a carrier for sickle cell disease, you don't have it, but you are a carrier, your blood actually becomes slightly sickled just a little bit, you are less likely to die of malaria. It protects you against, from mosquitoes spread that, it protects your red blood cells a little bit better. So if you think about it this way, if you do not have a sickle cell gene in your body at all, in your cells, you could die of what? Malaria. Okay? If both your genes say you have sickle cell disease, if you're homozygous for that, it's a recessive trait, then you will probably perish earlier in life due to what? Sickle cell disease. But if you're a heterozygote, you will not die of malaria and you will not die of sickle cell. It's called heterozygous superiority. You're more likely to stay alive, and that's why that stays so high in numbers, even though it can be lethal, you know, for those who are homozygous for it by the time they're in their 20s, depending on drugs, medication, and things like that, and what they're exposed to. Okay? So that is a recessive disease. Okay, so Tay-Sachs, we talked about that. Remember cystic fibrosis, the pounding on the back? Remember, that is also a recessive disease. But remember, you can wear a percussion vest and pound, pound, pound on your back, but it still will clog up all your other organs as well. And that's eventually why you would have a shorter lifespan if you had cystic fibrosis. Also, again, a recessive disease. Okay, and I listed those there for you. Do you see them? Okay, all right, now, autosomal recessive, so not on sex chromosomes, that's what we just talked about. Now let's talk about autosomal dominant diseases. So take a look here. Now, how does this look different from the other pedigree? Let me just jump back, look at this pedigree. Okay, how does this one look different? Discuss with your bio buddy.
The disease is a dominant gene, so if you have a baby,
Okay, so whatever this disease is, let me give you an example of one. Huntington's disease is a dominant disease. You're going to see it more often on the pedigree than a recessive disease, right? Because a recessive disease, if it's autosomal recessive, you've got to get 2 bad alleles in order to get this disease. If it's a dominant disease, you only need 1 bad allele, and you're going to get it. Okay? Also, what's something else that's different about this one than the recessive when you can tell like patterns when you look on the pedigree? Can you see anything else? Can you ever hide this disease? No. Can a brown-eyed person hide that they're carrying a blue-eye gene so it could pop up in their children? Yeah. Can you ever hide a dominant? No. It does not skip generations. Okay? Recessive disease, it can remain masked behind a dominant allele. It can remain masked for multiple generations. You're not going to like hide a dominant disease. Boom, it's out there. You're going to get it. Now, at what age you get it, that can vary. And for example, Huntington's disease doesn't strike into your mid-40s. So right now, all of you, any of you could have the gene for Huntington's disease. You would not know until you got into your 40s, unless you got tested. However, since it's a dominant disease, who could you look to see if you could even possibly have it? Yeah, if your parents don't have Huntington's disease right now, then guess what? You're not going to have it. They can't hide the disease from you, and then it pops up later in your life, because probably most of your parents are older than 40, and so if they're going to have it, they would already have signs of it. So if they don't have signs of it, you're not going to get it either. The issue would be is if you don't know who your parents are or don't know one of your parents, then that's when it could happen. Okay? You can get tested for Huntington's disease. It is a terrible disease. When it strikes, you can't stand upright. You'll keep flailing around. You can't sit in a chair or lay in a bed. You will flop yourself right out of that chair or that bed. You have lost control of your body. They literally have to tie them to chairs and tie them to beds. Okay? It is a very difficult disease to carry. And the sad thing is, if you don't know who your parent is, right? And let's say by the time you're in your 40s, probably have you already had a kid? Yeah, so you pass that gene on. But again, you can be tested for it. But it doesn't skip generations. You're going to see it, right? You know, it's... okay, so let's write this one down. So autosomal dominant disorders. Affected children will usually have an affected parent. Now, let me talk to you because the usually should bother you, right? In your mind, you should be pushing against that, going, what do you mean usually? You just said it doesn't skip generations. So this is what I want to tell you about penetrance and expressivity. Okay, so just listen, get your eyes up here so you're listening, okay? Penetrance is some certain genes, they'll say it has 70% penetrance, which means 70% of the time you express that allele and what? 30% you don't. So it could be you never express it. It could be hidden, it could be masked. It may have to be in conjunction with some other chromosome and collaboratively then it gets expressed. So that's why some alleles do not get expressed. You have 70%, let's say, penetrance or 85% penetrance. Expressivity is how you express it. For instance, you could get the trait for webbed fingers and toes, and you could have complete webbing between them like you are a really good swimmer, or you could have slight webbing, just a little bit of webbing between your pinky finger and your ring finger. Okay? So that is how you express it. For instance, if you know somebody who has Down syndrome, you know that there's a wide range of where they max out. You could have somebody max out at the 11 or 12-year-old developmental age, and they will, you know, communicate with you and speak with you and behave in a way which is appropriate for an 11-year-old. Then you have others that will only reach a developmental age of 4 years of age. Okay? If you look at being autistic, there is a wide range of autism. You could be on the spectrum yourself in here, okay? And you're just kind of touching it, but then there's all the way to the other extreme of autism. So there's a wide range of where you could be on that. So that would be expressivity, how that trait is expressed. Okay, so that's why it says usually has a parent who has the disease. The next one, number 2, heterozygotes are affected. Blue, could you please explain number 2, how heterozygotes are affected? And Slate, you explain 2 affected parents can produce an unaffected child, and use this pedigree to help you do that. Go ahead.
Okay, so heterozygotes are affected in this one. Since it's a dominant gene, if you have then you're going to develop that dominant.
Okay. Even if the letters were not written right here, if these letters were gone, but the coloring was there, you would know they were a heterozygote, right? If it was colored. Because you know they have the disease. Who did they get the disease from? Their mom or their dad? had to get the disease from their dad because their mom doesn't have it. So you know they have a big A because they have the disease. How do you know the second letter is a little a? Because they're mom. That's all mom has to give. Okay? So when you have an autosomal dominant disease, you're not going to mask it. You either have it or you don't have it. Okay? Over here, this daughter, what were the odds? What were the odds that she would have gotten the disease? She didn't get the disease, but what were the odds that this daughter right here could have gotten the disease? Could have gotten the disease. 75%. Right? And she beat the odds because what were the odds that she wouldn't get the disease? 25%. She didn't get the disease. Did she have to worry did she have to tell her husband when she got married, you know, this disease is running my family, you know, we maybe should go get checked because maybe we're going to have a kid? Would that be something she would need to tell her spouse? No, why not? Yeah, it's a dominant disease. This is why people want to find this stuff out. How is this inherited? Because she didn't get it. Even though both her parents have it, she didn't get it by the very fact that she doesn't have the disease, right? So that tells you her parents were heterozygotes. She doesn't ever have to worry about passing this particular disease on because it's an autosomal dominant disease. She doesn't have the disease to pass on. Okay? Ask your bio buddy. Easy peasy. You got any questions on that one? All right. Let's look at some examples of autosomal dominant disease. One is neurofibromatosis. There's lots of little birthmarks you can get, but these are little tan coffee, like coffee with cream kind of colored spot. And you want to keep an eye on these spots, especially if they're exposed in an area that would be exposed to the sun a lot, like your face, because they can turn into tumors. Okay? Not necessarily hurt you tumors, but be uncomfortable or hard to not focus on, I guess is what you could say. All right? So, want me to go back to the other picture? Okay. So let me tell you a story about this. I had a girlfriend, they had their first child, and the baby was born and she was talking, you know, had the spot right on the cheek, a little spot. And I said to her, as soon as she was talking about it, in a non-know-it-all, sciencey teacher way. I promise I said it nicely. But I said, which one of you has those spots? And she goes, what do you mean? And I said, well, one of you has a spot like that on your body somewhere. It wasn't her. She goes, how'd you know that? And her husband's name was Evan. And she's like, how'd you know that about Evan? And I'm all, you know, because his spot is located in a place that I would not have ever seen. Okay? And so she goes, how'd you know that? And I'm like, well, because, and then I said, it's an autosomal dominant disorder, so one of you had to give it to your child. And what they had to do is they had to do surgery to remove it when the baby's small enough so that that skin can grow back because they were worried that it could become a tumor like what you saw over there. Now I'm going to tell you, I had a student in my classroom who had neurofibromatosis right down the middle of her face. And it wasn't like, you know, a straight line. It was like this, went across. And it went across like this, and it affected her vision. You could tell she had it. And my heart was broken for her because there's not a surgery you can do to remove that much of your skin. That was just something she had to work with her whole life. Her one eye did not work as well as a result of it. And she had to definitely keep like sunscreen and things like that on it. So it can be at all different levels. So sometimes it's good to be thankful, like, you know, we get upset at ourselves or the way we look and just go, well, I do not have neurofibromatosis all over my face. You know, that is a hard burden to bear, okay? Because that is a dominant disease, okay? Definitely for her. Okay, here's another one. Huntington's disease, I told you about that. Uncontrollable movement. Again, it doesn't strike until you're in your 40s. Achondroplasia, the most common form of dwarfism, is an autosomal dominant disease. And so, could two dwarfs with achondroplasia have a non-dwarf child? Yes. Yeah, what are the odds? Yeah, if they're heterozygotes. But if one of them is homozygous for it, is there any chance? No. Okay, then this would be one of those issues that might surprise you. I would have students read about in bioethical issues. There are two dwarfs, married, pregnant, go into the OBGYN to talk about it, and they said, we want to have this baby tested, you know, decide if we want to keep it or not, or have an abortion to see if they're going to be a dwarf. So ran the test, came back, got the results, and they said, you'll be happy to know your child, you got the 1 in 4 chance. You will have a normal-sized child. And they said, oh, well, I want an abortion. And the reason was they were dwarfs, and they wanted to have a dwarf child. Yeah. So don't assume you know the rest of the story. And again, I'm not, #no judgment, okay? No judgments. But I'm just saying that's how it plays out. You never know what somebody, is trying for or wants, so don't assume you know the answer to that. I will only say as a preface to you, okay? I will say as a preface to you. I would tell you if I have not had an abortion, so I cannot speak to that. I never have. But I'm going to tell you that I have met women that do have it, and the age that it usually happens is your age right now. It's an unwanted pregnancy in high school or college, and they've got goals for themselves, and having a child right now is not the thing. Do you know how to prevent an unwanted pregnancy? How? You could, first of all, just not have sex yet. That way it turns out to be 100% effective. I know, it's crazy. Or use some sort of protection, like a condom, right? And the only reason I bring that up is because sometimes when you're in high school and college, you're just thinking about the here and the now. You're not thinking about tomorrow. You're not thinking about when you're my age. But I have met a woman who was crying in a bathroom, and women, like when we see somebody, we'll be like, can I help you? That's just typically how we tend to go. You guys will be like, I pretend I don't see it. Time to walk away. Okay? But women, we will not do that. We will be like, okay? And I went out and I'm like, are you okay? And it happened to be at church in the bathroom. And I don't know what had triggered it, but she had said that she had had an abortion, and she was so devastated by it, not at the time she got it, but now I think she looked like she was probably in her 40s at that point. And you know what the ironic part was, which was so difficult, was that she and her husband had been trying for 7 years to have kids, and they couldn't. And she was like, I always, and it was so difficult for her because she really, really wanted a child, and now she couldn't. And you're not going to say, oh, karma's a bitch. You're not going to say something like that ever, right? And I'm not thinking that. It was the irony of it. It just broke her heart. So I'm just telling you, I don't want you to be in that situation. I remember how she felt. I remembered the look on her face of it. And again, no judgments. I'm not judging you in any way, shape, or form, nor will I ever, unless you're mean to children or old people or animals. Then I will judge you, okay? But I would not judge you about that. But I'm just saying I don't want you ever to be in that situation I saw that woman in, and just the heartbreak that she was feeling over choices she had made in her youth. So make good decisions, right? That's something you have control over. Most of the time you would have control over that. Okay, next. Okay, is the next pedigree an example of an autosomal dominant disease or an autosomal recessive disease? So you're going to analyze that pedigree and decide what would give you evidence to say, you know what, if this family came to you with this information, you would say, I think this is an autosomal dominant disease, or I think this is an autosomal recessive disease, and tell the pedigree of number 8. I'm sorry, not the pedigree, the genotype of number 8. Okay, there you go. Have that discussion. They're whirlers. Yeah, you're right. Look at you. Sorry now. Okay, can I tell you how, come to your decision. So in your mind, I'm not asking you to say it out loud, just come to some decision in your mind. Have you got your decision? Okay, so when I get faced with a problem like this, right away, to decide if it's autosomal dominant or autosomal recessive, the first thing I know is if it's dominant, I'm going to see more of it in that family chart than I would if it's recessive. Do you agree with that? Yeah. So then I would go, okay, so if I see more of it, do you see more of it in here? Yeah. Then my first thing is, it's probably, more than likely, it's autosomal what? Dominant. Then you just kind of look for exceptions to it. You kind of play it out. We know autosomal dominant diseases cannot skip what? Generation. So anywhere up there, did it skip a generation? No. Then more than likely, it is an autosomal dominant disease. That's how I would approach that. Okay? Now, individual number 8. Let's go all the way back to coupling 1 up there at the top. Okay? Let's use the letter A just to work on this. If father 1 here has a disease, what do we know about him? Yeah, so he has at least one big A, right? That's all we know so far if that's all we have to go off of. What do we know about his wife? 2 little A's. Okay. Now, they had how many children? They had 2, right? Because their children not marrying themselves. They had 2 children. They had a little girl and they had a little boy. Do you know the genotypes of their little girl and their little boy? Do they have the disease? Yes. So you know they have at least what? One big A. Okay? Do you know their second letter? Yes. How do you know their second letter? Because their mother only had little A's to give. So I know they're big A, little A. Do I know yet what this second letter is? No, I don't know. Okay? What were the odds if they were big A, that all their children would have the disease? 100%. But how many children did they have? Only 2. That's not enough to go off of, right? If they were big A, little a, what were the odds that a child would get that disease? If they were big A, little a, what would be the odds that their child would get that disease? 50%. Because that dad would either give the big A or the little a. Little a, we're in the clear, right? Okay, I can't tell with only 2 children. That's not enough evidence to go off of. Okay, the only thing I could know for sure is if they had a 3rd child, or let's say he met somebody else who didn't have the disease, and that child didn't have the disease, then I would know he was big A, little a. You follow that? Okay, so now we know this son was big A, little a. What is his wife? Or partner, whatever. What is she? Little a, little a. So what's number 8? Big A, little a. Big A because she has the disease, and who did she get her disease from? Her dad. Okay, and we know her second letter is a little a because why? Her mom. Okay, check with your bio buddies, see if they understand that all right, if they have any issues over that. Okay, and then those of you who missed class, that was discussion 1. We're going to have one more discussion today, but I'm going to break it up into 2. All right, so hope we are making good choices.